What if we could edit life like we edit a document? Deleting diseases, rewriting our DNA, even designing the very future of evolution itself. This isn't science fiction anymore. It's happening right now, in labs all around the world. We've spent decades learning to read the book of life, and now we're finally learning how to write it. The tool that makes this possible is called CRISPR, and it's giving humanity the power to edit genes with a precision we once only dreamed of. It's a technology that holds the promise to cure our worst diseases, but also forces us to ask some of the most profound questions about who we are and what we want to become. To understand CRISPR, we first need to remember what DNA is. Think of it as a molecular code, a vast instruction manual written in just 4 letters, A, T, C, and G. Every cell in your body, from your brain to your big toe, carries a complete copy of this genetic blueprint, over 3 billion letters long. It's the code that builds you. But sometimes there are typos in that code. A single misplaced letter can cause devastating genetic diseases like sickle cell anemia or cystic fibrosis. For years, we've only been able to treat the symptoms of these conditions. But what if we could go back to the source code and fix the typo? That's exactly what CRISPR allows us to do. It's a molecular pencil and eraser for the typos in our genes. The story of how we found this incredible tool is almost as amazing as the tool itself. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, wasn't invented in a high-tech lab. It was discovered in bacteria. For billions of years, bacteria have been at war with viruses. To survive, they developed a clever immune system. When a virus attacks, the bacterium captures a small piece of the virus's DNA and stores it in its own genetic code, like taking a molecular mugshot. If that virus ever shows up again, the bacterium uses that stored DNA to recognize the invader and deploy a special protein to slice its DNA apart, neutralizing the threat. For a long time, scientists saw these strange repeating sequences in bacterial DNA, but didn't know what they were for. Then a light bulb went off. Researchers, most notably Jennifer Doudna and Emmanuel Charpentier, who won the 2020 Nobel Prize for their work, realized this ancient bacterial defense system could be repurposed. They understood that if you could give this system a mugshot, any DNA sequence you wanted, you could guide it to cut DNA at a precise location in any organism, not just a virus. Suddenly, we had a programmable gene editing tool. So, how does it actually work? Imagine you want to fix a typo in a giant book. First, you need to find the exact sentence with the error. That's the job of a molecule called the guide RNA. It acts like a genetic GPS, programmed to find a specific sequence of DNA letters. Once it locks onto the target, it brings along its partner, a protein called Cas9. Cas9 is the molecular scissors. It cuts the DNA at that precise spot. At this point, the cell's natural repair machinery kicks in to fix the break, and this is where the magic happens. Scientists can trick that repair system into either deleting a small piece of DNA or, even better, pasting in a new, corrected sequence that they provide. It's like the find and replace function in a word processor, but for the genome. CRISPR doesn't just edit genes, it edits possibilities. The applications are staggering, and they're already moving from the lab into the real world. In medicine, CRISPR is being used in clinical trials to treat genetic disorders that were once considered incurable. For sickle cell anemia, doctors are editing a patient's own blood stem cells to fix the mutation, and then reinfusing them. Early results have been life-changing. We're seeing new cancer therapies where a patient's immune cells are edited to become superpowered cancer hunters. And there's research into editing our own cells to make them resistant to viruses like HIV. We've moved from just managing symptoms to fundamentally rewriting the code of disease itself. But it's not just about medicine. In agriculture, CRISPR is creating crops that are more nutritious, more resistant to drought and pests, and can grow in more difficult climates. This could be a huge step towards feeding a growing global population. And in environmental science, we're exploring ideas like editing mosquitoes so they can no longer carry malaria, or engineering bacteria to clean up oil spills and other pollution. The potential seems almost limitless. But with great power comes terrifying responsibility. And this is where the conversation gets complicated. So far, we've been talking about editing the cells of a single person to treat a disease. The changes die with that person. But what about editing human embryos? This is called germline editing, and it's a completely different ball game. Any changes made at this stage would be passed down to all future generations. In 2018, the world was shocked when a Chinese scientist announced he had done just that, creating the world's first CRISPR babies with the goal of making them resistant to HIV. The backlash from the scientific community was swift and severe. This event threw the ethical questions into sharp focus. Who gets to decide what genetic traits are defects to be fixed and what are just normal human variations? Where do we draw the line between healing a terrible disease and enhancing a person to be taller, smarter, or more athletic? Could this technology create a new form of genetic inequality, a world of the genetically enhanced and the unenhanced? The same tool that could end inherited disease could also fundamentally redefine what it means to be human. And now there's another powerful force entering the picture: artificial intelligence. AI is supercharging the world of gene editing. It can analyze massive genetic datasets to predict the unintended, off-target effects of a CRISPR edit, making the process safer. AI algorithms can design more effective guide RNAs, increasing the precision of the cuts. We can now use AI to simulate the effects of a genetic change in a computer before ever trying it in a living cell. In labs all over the world, AI and CRISPR are now working hand in hand. Intelligence is meeting biology. We are witnessing the merger of two of the most powerful revolutions in history, the digital and the biological. The technology is also evolving at lightning speed. CRISPR-Cas9 was just the beginning. Now we have next-generation tools like base editing and prime editing. These are even more precise. Instead of making a full cut across the DNA, they can go in and chemically change a single DNA letter into another, like a true pencil and eraser. We're also developing gene drives, which can spread an edited gene through an entire population of a species in just a few generations. This could be used to wipe out invasive species, but the ecological consequences could be catastrophic if we get it wrong. One day, we might cure all genetic diseases, or we might accidentally change the course of evolution forever. The question is no longer just, can we, but should we? For all of human history, we have seen life as something sacred, something given, something to be observed and understood. Now, for the first time, it has become editable. The code of life is now a programmable medium. CRISPR shows us both our own brilliance and our blindness, our incredible hunger to create and our desperate need for wisdom. We are no longer just the products of evolution. We have become its editors. Thank you so much for watching. If this journey into the future of our own biology fascinated you, please consider liking this video and subscribing to the channel for more deep dives into the science and technology that's shaping our world. See you next time.